So I just wanted to also acknowledge that we have 100 people or so online. So online people, we're going to start up again. I want to introduce Dr. Brad Britigan, who is the Dean of the College of Medicine here at UNMC. He's been a great supporter of research and integrating it every way possible into the clinical mission. It's not so easy these days, but he's given us a lot to support research through things like the Dean's Development and Initiative, DDIF, which I forget, Needs Discretionary Investment Fund. That was the word I was having trouble getting. But it's very helpful. He's helped with startup packages. He's been extremely helpful in getting us able to remodel space for AI and machine learning and clinical research informatics, and for an attendant cluster hire. We're already starting to bring folks here. JoJo is here. Hello, JoJo. She's the camel's nose of the tent, and we have hopefully many more on the way. Dr. Britigan is a colleague from way back at the University of Iowa. He spent time at Cincinnati, and he's been here for maybe 11 years or so. Time flies when you're having fun. Yeah. And so Dr. Britigan is going to introduce Dr. Phil Polgren. So thank you.
Thank you, Matt. I appreciate the invitation to introduce Phil, who I have had the pleasure of knowing for 25 years, dating back to our time at Iowa. Obviously, as the slide lists, Phil is professor of internal medicine in the Division of Infectious Diseases at Iowa. And I first got to know Phil in my role as, at the time, the division director of ID and the fellowship program director at the time, as Phil came to Iowa as an internal medicine resident and then an infectious disease fellow after completing his MD degree at the University of Cincinnati in undergraduate training at Beloit, where he was a math and economics major. And as I got to know Phil during his fellowship, it quickly became apparent that he looked at research opportunities and challenges differently than many individuals who traditionally have matriculated into infectious disease, and really calling upon his interest and background and expertise in mathematics and computer science to think about novel ways to approach these problems. Now, I left Iowa to go, ironically, to his alma mater, medical school alma mater in Cincinnati in 2004, just as Phil was joining the faculty at Iowa. But I would frequently see Phil probably annually at the infectious disease meetings, and we would also always find about 10 or 15, 20 minutes to talk about what he was doing. Usually during the course of the conversation, as Phil was telling me about his ideas and concepts, usually the phrase, "Does this seem crazy to you?" came up. And usually my first response as he was telling me about what he was thinking about was, yeah, it does seem a little crazy. And then the more I thought about it, it was really novel and incredibly creative. So the whole concepts around computational epidemiology, which is the direction Phil's career has gone, and has been very successful with well over 200, I think now approaching 250 peer-reviewed publications, numerous grants, numerous students he has trained, has been a testament to how he has viewed and really kind of created that field. And I'm looking forward to hearing his talk today, which you'll be talking about basically using real-world data to inform clinical research. So, Dr. Polgren.
Thank you. Thank you, Dr. Britigan. That's I kind of thought you thought some of the ideas were a little bit out there, but I tried not to include too many of those today. So hopefully these are all not too crazy. Just I think they're just things that I generated just from seeing patients in clinic. And you know, I'm glad the term real world data came up, because before that I did stuff and I just said I just do stuff with data that kind of fell off a truck or something, you know, just stuff that was just there. But now there's actually a word for it, or a phrase, real world data. Before I get too far, I should say I don't have any disclosures that relate to this talk. Yeah, real world data, it's you know, it was this term was coined a few years ago, and it really applies to kind of non-structured, well structured, but data that's not kind of lives outside clinical trials. It's it could be administrative data, it could be patient collected data, it could be data from electronic medical records. So it's usually observational data, but it really there's more and more of it, and I think that last talk sort of showed some of the promise. You know, Matt asked me to give this talk, and I just love Omaha, so I thought this would be great. And he said, "Well, what are you going to talk about?" And he said he wanted me to talk about real world data, and you know, I didn't know what that would be, so I took this slide from there's a CPSA meeting, and they're going to talk about real world data, and they have this outline. Let's talk about clinical data, patient generated data, cost and utilization data, and public health data. And these are all breakout sections. So I thought I'd just combine them all together. And then if someone doesn't like part of the talk, you know, if you wait a few minutes, maybe there'll be something else that you like. So I'm just going to show a bunch of different projects, kind of cases of real world data that we've done at the University of Iowa. Some using administrative data, kind of non-standard uses, some data from devices, some data from kind of patient reported data, and data from kind of non-traditional devices that are just already out there. Maybe you could get at the hardware store or a retail store that aren't designed for health data, but that you could use. So there are a wide range of projects. I'm an infectious disease doctor. Thanks to Dr. Bridigan, he got me interested in infectious diseases. And all of them are pretty low budget. This is sort of a fun on a budget. I don't have a company or anything else. And I always put the project before the platform. So the platform should, you know, serves the project and not the project serving the platform, if that makes sense. They all involve multidisciplinary teams. And that's not by kind of design, it was really by necessity. They've all involved trainees or students. Every single project I'm going to talk about, either a medical student, a graduate student, or a trainee, like a resident, was involved. Most result in publications. A lot of them were discouraged before I started them. Like, why would you do that? Not all. And some are successful, but there were some failures too. And I probably should have had more failures. And I probably learned more about those. But all these projects you could do here, you could do this at any Midwestern university. You don't need a lot of resources. The information is there. You just need to use it to answer some clinical questions. So I'll just start with administrative data. A lot of people probably in this room have used administrative data. So HCUP data or Optum, or maybe does your program support that? Maybe the medical school has, you know, supports the use of this data. It can be private insurance data. It can be public insurance data like Medicare, Medicaid data, claims data. And this has been used a lot in something called health services. And here's an example of a paper, and I wish I'd had this idea. It sort of shows that, you know, surgeries are going up over time, and that was in JAMA. And, you know, I never thought that could, I guess I wish I would have done more projects like that, but I've never had a publication in JAMA. But that's like the standard health services. You know, you plot something's changing over time. People are using services in a different way. So I've been using MarketScan data, and it is a little expensive, but before that I used HCUP data, and I used some projects, it was like $35 to download all the hospital administration, like all the hospitalizations and ER visits in California. I think Arizona at one time was $20, and if I had a student buy it, it was even cheaper. The MarketScan data is a little bit more expensive, but it has outpatient visits, ER visits, hospitalizations, outpatient drugs, and it has sequential visits. So unlike a lot of hospitalization data, you know what happens at your hospital, but what if they go to Lincoln? You lose that. And so the thing I like about this is it's sequential, so you can follow patients over time. And there are millions and millions of patients. So I'm going to give you a couple examples of some stuff that you can do. So a lot of people are talking about climate these days. And so one of the things we've been doing is taking market scan data and then fusing it with other data. So it turns out for free, you can get weather data from all the weather stations, so like hourly, precipitation, temperature, wind speed, all that is available. And so we just downloaded it and then matched, not all of there's not a weather station in every sort of zip code, but it's close. And so we basically built a database so we can actually compare over like the last 20 years. We can't study climate, that's more long-term, but we can study weather. And we can look at that, and we started to study how weather can affect health. And so I'll give you an example. One of the first things we did is we were interested in deviations of temperature. So let's say unexpectedly warm days where it's usually cold and vice versa. And so, we started building these graphs and then figures and then overlaying clinical data. And one of the things that I was interested in is urinary tract infections. Is one of the most common causes of a visit for some people for infections, like one of the top 3 visits in outpatient clinics. And, it turns out they're seasonal. I didn't know that, but a very observant Canadian found this in about the 1980s and published a paper showing it was very seasonal just in his clinic. It's been replicated in Greece. And we started writing some papers showing just in HCUP, which is very cheap, that it's very seasonal. So they spike in the summer and they go down. But what we wanted to do is look at weather. And so that's exactly what we did. And here we looked at the incidence of UTI visits by temperature. We looked at the temperature the 7 days before. And our hypothesis is that it's really driven by volume depletion or dehydration. And some people would say, well, maybe weather's correlated with other things, maybe it's just the summer, but it turns out we can look at every single month and we get the same pattern. So this is something that really wasn't well known even by urologists. We published this in the Journal of Urology. It wasn't really known by nephrologists, and ID is not really widely appreciated. So we discovered sort of a novel risk factor, kind of fun on a budget. It's done very easily. Half the data was free. So we've done this with several different infections and showed that really there is an effect on the environment. And in some cases, it can be a major risk factor. I'll give you another example. I never thought that appendicitis would really be driven, but I read a paper about seasonality. And indeed, if you look at the incident ratio, as the temperature goes up, the risk of an appendicitis goes up substantially. And I don't know, I don't have a mechanism for this. It's probably due to volume status. There have been a couple papers about seasonality in different parts of the world. But this is a project that we were able to do very easily with just sort of, data that was just sitting there already. Learning about a new risk factor for a very old disease. We looked at surgical site infections. And here, it was very seasonal. We wrote probably the 3rd seasonality infection, strikingly seasonal. And it's not because the surgeons just take vacations, in the winter. One of the reviewers thought about that. We compared it. We again mapped in temperature data, and you can see that the risk or the odds of an infection go up substantially as temperature goes up, and you can just see that it's almost a linear dose effect. And again, we have some hypotheses for why this is. It's true actually for cellulitis as well. The risk of having their cellulitis, especially the legs, is extremely seasonal. Might have to do with leg swelling. We're still working this out. But this has generated a lot of questions. There are possible mechanisms. But for surgical site infections, the degree of risk is similar to sort of summing up comorbidities. So it's really well established that people that are overweight and diabetic have at risk for surgical site infections. And the risk that we see kind of comparing like Amarillo, Texas to Maine, that's about the amount of risk that you see from some of these patient-related factors. So again, very easy to do. Anybody could have done that. These are great projects to do with students, trainees, et cetera. And that can sort of answer questions and let you start studying the effects of climate very quickly. All right, here's another example. We've been using this to study diagnostic delays. So diagnostic delays are one of the new Diagnostic errors are one of the new things that HRQ has been talking about as the next frontier of patient safety. And if you think about it, it's really important. And we've been using the same database, but we combine that with some models and some simulations to actually figure out how long it takes to diagnose someone on average. So I'll give you some examples of why we should do this. So I'd say my claim is that diagnostic delays are one of the most important forms of error. Well, it's one of the major causes of lawsuits, but that's important to think about. But also, it really encapsulates all the other errors. If they're errors of like wrong drug or wrong test, the way those are going to manifest in taking longer time to diagnose the patient. So it sort of is a summary of others. It's easier to measure. And so in general, if you delay, you'll delay the treatment. But even if there's not a treatment delays can cause anxiety in patients and families and can put a health on the stress on the healthcare system. If you think about it, there are all these visits, extra visits that are used. We'll never get those visits back. So one of our kind of themes is trying to increase diagnostic efficiency. And so we've been using this data, this real world data, to just sitting there to sort of study these things. So the reason to do that is nobody really knows the incidence of delays. When I got into this, I could not figure out, there's no really good papers about what disease is most missed, how long does it take, what are the risk factors of delays, and what are the consequences. They're kind of, you can imagine consequences. And maybe, there are really no metrics. And so if you have a metric, how are you going to sort of study this and measure this and try to do an intervention? And so, studies to date have been really hard. They're they use autopsies or lawsuit cases. They're usually in sort of large medical practices or hospitals, but not ambulatory settings. So if you have this continuous data, our claim is you can use this, again, this real world data. So what we've been doing is we've been taking the sort of kind of a graphical approach. And this is a paper we wrote on TB. This is actually what we observed from about 10 to 15,000 cases of tuberculosis. Not surprisingly, we plot all of the symptomatically similar diseases. And if you can see, I think this arrow works here. This is a laser. Oh, here we go. Okay, so you can see that about 100 days or so, the visits start to go up. And these are symptomatically similar diseases. So what are those? Cough, shortness of breath, asthma, pneumonia, not TB. As you get closer, this is the index date. And you can see all these extra visits start to accumulate after about 100 days. So these are probably coincidental symptoms because people have cough without having TB. And as you get closer to the time of diagnosis, it starts to go up. It's about 100 days. So we use something called change point, a time series method, where we can actually kind of estimate this window. When does it start? These are probably missed opportunities. And this is probably, sort of expected levels of care. And so what we can do is these are probably the missed opportunities in blue, expected unrelated visits. And we can actually, we don't know which are which belong here, which belong there. But if we use this data and do some simulations, we can actually figure out what are the chances that any of these patients experience a diagnostic delay, what the length of that delay is, whether a specific delay kind of lasted a specific period of time. And then with that, we can actually discover risk factors. So we can actually look at the simulation and see what factors help drive this, what presentations help drive this, what comorbidities help drive this. So I'll just give you a Dr. Rizzo's here. So here's a neurology example. So this really surprised me. This is herpes encephalitis. We've done this with tuberculosis. We've done this with histoplasmosis, pertussis, but herpes encephalitis. Let's see if okay. So I was really surprised by this, but it looks like these SSDs or symptomatically similar diseases start 28 days out, which seems like a really, long time. But indeed, I went back and I love doing these projects because I get to learn about these diseases that I should have known about already, Dr. Diggins. Sorry, but You know, every time I write a paper, I learn more. And it turns out that you go back and look at case reports, and there are indeed cases of subtle cognitive findings that develop that far back. Now, they don't all have, they probably have word finding difficulties. They're confused. They don't all seem, you know, they don't all have fevers and seizures. But as this goes up, so what we can do is we can actually look at the delay. The average delay is 5 days, but there's a wider window, and we can put confidence intervals in this. We can look at the average numbers of missed opportunities. It's like 2. That means this is a life-threatening disease, and people are being sent home from the emergency room routinely. And 11% of patients had about a 2-week delay. And then what we can do is we can look at individual risk factors. And so we can actually look here, age less than 65. People think about it in older people, but our hypothesis is that they don't in younger people. This is like a cognitive bias, this sinusitis or schizophrenia. So if someone comes in and has a history of sinus infections, they're more likely to be sent home because they have a history of having headaches. And clinicians, I learned this from one of my friends in medical education. They have an illness script and they prematurely close and they kind of move on. They sort of, here, I'm done, have a sinus infection. And then schizophrenia, people also ascribe all the kind of mental status changes to that pre-existing disease instead of actually having herpes encephalitis. Yeah. Yep. This is the 10,000, or in this case, it was about maybe, I think we had 8,000 or 9,000 cases. So each time we do a disease, we build a cohort. So we're doing one with sepsis now. I have like 200,000 patients. I've done it with Q fever with only like 300 or 400 patients. And the bigger the cohort, you know, the better our estimates. We're using Market Scan. We spin up a big cohort and we basically go back and look at this administrative footprint, this sort of path or diagnostic odyssey of 10,000 people. And from that, we can kind of see what are the missed opportunities, the averages, and then we can go back and look at the individual patients. I'm not sure. I don't. So at any rate, I can go back and do this for other diseases. And if there are people in the audience that are interested in particular diseases like Parkinson's disease, you know, we can do this. If you're looking, we've done it with strokes, heart attacks. It's not just for infectious diseases. They're great because they're acute and timely. So I'm focusing on it, but it can be used for any diseases again. And it can actually, you don't need to use Market Scan data. We've done this with HCUP data that I talked about. We're working with Kaiser Permanente, where we actually not just have administrative data, but we have lab data and vital signs. And we can kind of look at that. We can also look at the presenting factors and features. So what if someone has a fever or doesn't have a fever? It turns out, we think, and I think I have some preliminary data, if someone comes in and doesn't have a fever, they're much more likely to be missed because that illness script is kind of running. And if there's no fever there, people just don't start going through their kind of, all good clinicians are Bayesians, and they don't sort of generate that kind of list, and they don't even think about it. All right, so that's kind of the second example. And if you don't like that, like I said, I'll just keep moving. You can use this for genetic diseases. Now, this is kind of a weird thing, like using administrative data to study genetic diseases. And I'm not sure you can use it for more than one, but I just had that example. I'm not a genetics expert at all, but a lot of people at Iowa study cystic fibrosis, and I don't know much, but it's an autosomal recessive disease, which I have to think about that, which means you need 2 copies. So, you know, we were studying kind of cystic fibrosis, and it's a disease. It's autosomal recessive. It causes hyperviscous mucus, a lot of respiratory infections, sinopulmonary infections, but it affects a lot of other organ systems. It's a sodium chloride channel. And carriers have half as many channels that work. Turns out that 1 out of every 33 people in Iowa and Nebraska are a CF carrier, and they don't know it. So one of our questions was, I just didn't know any better. And I was starting to look at the health effects of being a CF carrier, having just 1 copy. And so I knew a little bit about genetics, and I thought, well, gee, if you find the kids with cystic fibrosis, you can look at the parents, and we can look at the parents' health. And I'm like, well, you know, if mom and the baby are in the hospital at the same time, probably mom's mom, unless there's a baby slip up. Not sure about dad all the time, but we can look, we can trace this. And through insurance, you know, in this country, you can kind of find those linkages. And so we could do that. It also turns out there's actually a carrier code that's generated. And I started this project, and then someone said, this is not a thing. And here, this is from Kaiser Permanente, and it says, being a carrier does not affect your health. And actually the CF Foundation has similar language. They think they might have changed it by now. This was Access 2020. And so, but screening's done. Actually, newborn screening, every kid is screened for CF. And it turns out we find some carriers when this happens. And then the other way we find carriers are through prenatal screening. So that allowed us to build a really big, a relatively big cohort. And we first started by thinking about CF-related diseases, like what diseases are CF-related. I reviewed about 1,000 papers and came up with a list of 61 conditions. And then we built this big cohort based on claims. Did they have a carrier code? And then we did a validation cohort looking at mothers, right? So the moms of kids with CF. And we found that basically every single disease that you find in cystic fibrosis, people that are CF carriers are at risk for. Not as great, but every single disease. Well, okay, we didn't find it for, I think it was like 50, 57, and 61 conditions. But our group is young because of the way we find them through newborn screening in mothers. And so we didn't find cirrhosis, which is actually very common among people with CF. A lot of them end up going to get liver transplants, but we did find elevated LFTs. We found jaundice was elevated. So if we think we had an older cohort, we could find all of these. So we've gone on. So I'll just give you some example. This is what I kind of wrote a grant about, all these infectious disease risks. All these infections that are so common with people with cystic fibrosis, carriers are at increased risk for. And in some hopefully soon to be published work, you can actually figure out, we've actually found that people with bronchiectasis seem to have worse disease than non-carriers. People with sinusitis have worse disease. And how do we know worse? Well, they're more likely to have sinus surgery. They're more likely to have more antibiotics prescribed, et cetera. And so it seems like in the world of bronchiectasis, there's actually a different phenotype in carriers. So it's a heterogeneous disease. So we think carriers sort of lie somewhere between the rest of the world of bronchiectasis and people with CF. And it has important implications for treatment now because there are drugs that can treat cystic fibrosis. And in some cases, they may help CF carriers. So that's my claim that you can actually even use, like study genetic disorders using the real world data. What about data from the internet that's just kind of out there? So beyond administrative data, I wrote some early papers, then I just got out of the way. I wrote the first paper using search terms. I worked with Yahoo Research at the time. At this time, they had a longer history than Google. And I met some people at a party, and that was a good party. And we planned this paper, and it turned out I had this hypothesis that, and I think other people did, I just had the data, that searches for flu kind of measure or sort of or proxy for activity. There's this interest that sort of perceives the actual kind of disease activity. Not too long after, Google Flu Trends came out. I couldn't really compete with Google, but a few years later, we did the same thing with Twitter. We had access to the pipeline, and we showed indeed the same thing, that tweets were kind of a early indicator for activity. And we were able to do this during the pandemic in 2009. But that's really crowded, so I got out of there. Lots of papers written still using search terms. There's really no barrier to entry. So a lot of people were doing this, so I had to move on. I couldn't figure out how to fund it. But anyway, there's a lot of information out there. So I started moving on to devices, and I wrote those papers, and I got a call from a guy who had a temperature, or like a thermometer company. He said, I want to predict flu. I'm like, okay. And he said, I have this company, I'm selling thermometers. And I said, well, I think you need a couple 100,000 thermometers. You need more than just a few. So I'd forgotten all about it. He calls me back. He said, I've sold 500,000. Is that enough? And I tried not to be super excited. I'm like, well, we could look at it. Okay. And so he sent us all this information. And And it's not just temperature data, it's geocoded. So we actually had timestamp temperatures. And because there's a companion app, we knew something about the age. It was usually reported, but we had age, we had timestamp, we had geolocation. And so what we could do is actually learn about a disease from this free data that I got from a company. Turns out he gave us 10 million readings across the study period, and they're febrile episodes. So this was kind of labor intensive, but we defined all these febrile episodes. And one of the cool things is this is at home. We know a lot of papers, we know a lot about temperatures in healthcare facilities, but what about at home? And so we realized this was an opportunity to sort of explore kind of how this notion of the bimodal temperature distribution. And so here's a paper we just published last month that actually shows that fevers and temperatures really change throughout the course of the day, and that's been known. But what we were able to do is kind of look across the age ranges and time and estimate the probability that someone would have a temperature at a certain time of day. And so this isn't surprising. You know, anybody that does geriatrics should know that, you know, this sort of common wisdom that older patients might not mount a fever. We actually see that. We see differences across the age group. Really, really big differences. But what's not known and appreciated is you can see the probability of having a fever. And what this means is that if you come in the morning, you might not have a fever. If you come in, that same patient, if they come in at 4 o'clock, they're much more likely to have a fever. So your chances of catching that really matter. And so we were able to do this with just data that someone gave us for free. COVID happened and he doesn't give us data for free anymore, but we still wrote this paper and we had to move on, and now we have to find another data source. But it just shows you what you can do. But that's not all. We wrote some flu papers too. And it turns out you can do a really good job of predicting flu from temperature data. And we can actually go to like the state level, which we couldn't do with search terms because it's so granular. And not only that, we could learn stuff about flu. So it turns out there's sort of this biphasic illness where you get influenza and then you can get a bacterial infection afterwards, like sinusitis or pneumonia is classic. And when you go to the old flu chapters, it's not in the new ones, but if you go back maybe a couple versions, they describe this and say it happens in maybe 2 to 5% of people. And so what we actually did is we looked at the seasonality of biphasic illness, or biphasic fever patterns, and it actually mimicked what the kind of old books said. You know, we saw about 2 to 5%, and actually they peak right when flu season does. So this is just the plot of biphasic fevers, where you have a fever, you get better, and have another fever. And interestingly, we can also look at the directionality, because it's like, here's the parent putting their fever in, here's the kid, and it's actually always kid, parent, kid, parent, kid, parent. And then the next thing you know, it's like kid, mom, kid, mom, kid, mom. Very rarely is dad. That my wife thought was very interesting. So you can learn about kind of household transmission from this. Okay, what about patient user data? Okay, this is data that Joan talked about. There's a lot of patient generated data, and you can use that. A lot of people have these phones, and this is a great new opportunity. We sort of have a different approach. It's kind of a fun on a budget. I don't have a big team. I don't have a company. And we sort of think of the project first and then see what do we need to do to finish that. And I think it's worked well for these projects where we can pivot from project to project without building an infrastructure or building an app. We tried that early on and got burned. It's hard to install apps. I found personally in old clinics, with older patients and such. And so we've been using kind of text messaging. I'll give you some examples. We wanted a scalable approach that was super cheap, pennies on the, just really cheap. And so that's kind of what we've done. We've been using text messaging to build an engine. And it's not so novel now, but it was 10 years ago when we first built it. Can we envision writing a prescription with a text message? Here's the message, you write it out. This is what I'd like, sort of like how often, twice a day, BID, and then the duration. And then we can get information back, and then we can actually it's bidirectional. So we get a message and we can send another message out. So sort of almost have a conversation, super cheap. You don't need an app. And if you can text, you can do this. So So this has sort of opened the door for collecting data on blood pressure values. My wife wrote a grant or has an R01 looking at rural patients, and pharmacists have been using this. And we basically, the goal is to try to control try to control blood pressure before that patient comes back to the clinic, because we wait, blood pressures are high, wait till the next clinic visit. So the idea is to get these kind of 14 measurements and start the drugs in a collaborative practice agreement before it actually ends up. But that's not all. We can do messages, but we can also pair it to any Bluetooth device, and we can get that API. It's not that hard. We've done this with Fitbits. We've done this with indoor pollution sensors, and we can pull the data down and get that kind of device level data and then send messages based upon the readings. We can do it with Bluetooth thermometers. Hey, I've noticed you haven't been wearing your Fitbit. Send that message, pull that back, have that directional. And we can also do this with MMS, pictures, photos, voice clips, etc. And super cheap. And my claim is this is a great way to do a project with a fellow or student. It doesn't cost any more money. And you can get that preliminary data that you need, or you can do that pilot project. So here's an example. We've done this blood pressure stuff. I've already told you about my wife's grant. Made her happy. I helped her for once. She's really helping me. So she used this engine to get this grant. I'm sorry, so it was an R01. It's an R6133. We can get photos. So I've really always been interested in diabetic foot ulcers. And I tried to build an app. It was called PhotoFoot. And I needed a large corpus of photos so that we could actually identify a foot. It's kind of hard to find on the internet. So I went out and got it. And a bunch of people sent me, I tried to pay them. They sent me a bunch of pictures of feet with their socks on. So, so we went to the clinic and started over. We worked with some podiatrists, got all these photos, and we had to get the ulcers too. And so we have one of the largest corpuses of diabetic foot ulcer photos, I think, in the country, thousands of them. And we've been building this. And then first we wanted to say, hey, can you send us pictures of your feet? So we did a randomized control trial, seeing how we could do it. We gave a third of the people selfie sticks to see if that would work. We had a third of the people take pictures on their own. That didn't work. And we had a third of the people have another family member take the photo. And about 95% of the photos, according to my orthopedic collaborators and ID ortho collaborator, would probably be helpful. They could see something that was helpful. It would be useful to them. And people were willing to do this for 6 weeks. And this is kind of what happened. People would take this. Now, some of the photos were surprising. This lady, we blocked it off. She was smoking, using her selfie stick, taking a picture of your foot. We saw some stuff we didn't want. So we built a kind of a pipeline. We don't want to see that. There's some other stuff we didn't want to see either. Now, some guy sent us pictures of baby deer. He would just send us photos from his yard. So we built a system to sort of automatically filter, black everything out. All we want to see is the foot. So we built that pipeline to recognize the foot. And then we annotated all the ulcers. So now we can find an ulcer. I have a collaborator that did this work. Talk about teams. He won't look at the photos. He thinks it's too gross. He had to give a talk because we got a pilot grant, and he wouldn't look at the slides. Next slide, because it was so gross for him. But he was able to help us do this work. No. And so here you can see in green, we can actually find that. And then I had the idea of putting a Scholl's, Dr. Scholl's insole, because, if you take it, how do you know how big it is? Well, if we know how big that is, the marker, and we can find 2 circles, we can identify that, and then we can actually size it. So we can see long distance for super cheap. We can tell if your foot ulcer is getting bigger or smaller remotely. And that's kind of, we're building that. We write the paper, and then we'd like to do something with this because we think it's a really cheap way of doing this. But that's not all. We had problems with texting. Some people were making mistakes, and I kept getting parking tickets, and I didn't know how it happened. It turned out they were taking pictures of my license plate and giving me tickets. The ticket wouldn't even show up. I just get the bill. And I thought, well, why don't we have people just take a picture of their blood pressure cuff? And so indeed, it works pretty well. So we actually have people take a photo and then, hey, send me a photo of your blood pressure display. And then we can use optical character recognition and then find out what their blood pressure is and then read that. And it's almost the same as a text. So one of the projects now that we're doing with the CTSA is we're comparing texting versus Bluetooth versus just taking a photo. The nice thing about the photo is there's actually in most of the photos that we're using, there's a timestamp in there. So we know people aren't just taking the same photo and sending it to it. So it's time and date stamped. So there's some sort of fidelity with it. I said we could use a newspaper headline or something like that. And my students were like, what's a newspaper? No one has a newspaper. So it works pretty well. And we're rejecting some of the images. It's usually due to lighting or angle, but we can, if we're unsure, we just reject the image. So that's like an area of investigation. We just finished an R21 with OPAT. You know, does everybody know what OPAT is? It's home therapy antibiotics. So you get a PICC line, you go home for 6 weeks. And so the idea was to take the hospital home. So we asked people, are you having a fever? Are you having any problems? And we can actually either use REDCap for this. So this is something you could do because REDCap has Swelio integration. You could send these questions. People could push, the question or not. We gave them a Bluetooth thermometer. Turns out they took their temperature like 15 to 18 times a day. So we actually stopped that and had them just enter it in. It was a disaster. People kept pushing them, just taking their temperature all day. And we had them take a cell phone photo of their wound site. That didn't work. They kept sending pictures of their bandage again. But so, we failed a lot, but most of the people were happy to do this. They were happy to say if they had any problems. And we didn't detect many fevers. We did this here at University of Iowa, not here. We did it at the University of Iowa, WashU, 2 very different populations. It was in our, and it worked. We were able to sort of touch and contact these patients remotely, and they were willing to do it. You can also do interventions. So when we've married our text messaging machine, we built a game. We've married it to like a Fitbit integration. We've kind of integrated that so we can tell how long people walk. And then we had this idea where we put the steps on a map. And since Fitbit, I had a long talk with them and I realized we couldn't compete. And they've integrated a lot of the stuff in sort of their group walks and stuff. But we had this idea that you kind of competitively walk, and as your little stick figure would be projected along the course as you took steps. So every time the Fitbit synced, your little character would go along. And it's like a soccer, our vision was like a soccer league. So you'd go every week, you'd go up or down. So we'd always be walking with like folks, so you wouldn't be blown away by someone who's super fast. And sort of the idea was to gamify this. Again, we did this. We didn't have a huge budget. We built this. It was all based on a text message. You get a text message. You could click it. You could see where you are on the map, or you could see like Google Street View. You'd actually see where you'd be walking if you were really walking and look around. It turns out over a Fitbit alone, we were able to get people to walk substantially more on average. You can see kind of the differences. And on average, over a Fitbit alone, we got people to walk 81 miles. So it was kind of ultimately a failure because it went away, but it might be just the ticket for someone who had a hip or knee surgery that you really want to have walk a little bit more. So again, we added this game, and over just getting a Fitbit alone, we got people to walk 81 miles more on average. All right, so for people that kind of another version we built inside a long-term care facility. So in this case, we wanted to encourage socialization. And so people got on teams and everybody was able to see what color they were. And so the idea was that people would form teams. We didn't want to be hugely competitive. And so we wanted to be a social event. And every time they walked by a certain part of the residential care facility, all their data would be downloaded on Poisson, and they'd see kind of their team versus everybody else's team. And they could kind of socialize. It was working really well. And then this thing called COVID happened. But we were still able to, show that it worked. People liked it. I'm going to go over, we've already talked about Parkinson's, but we've been kind of getting audio files to try to look at tremors remotely. Super cheap, super easy to do. Just send a little video clip just by text messaging. We've been analyzing that. Let me tell you about a huge, a failure. This didn't work. Maybe it was the name, Suit Yourself. That was one of the ideas. We had a surgeon that liked to operate, but he did not want to see patients after, so he said, this is what we need. We need something like a bidirectional system that teaches the patients how to take out their own stitches so I don't have to see them. And so we picked doing this with carpal tunnel release, and we had them watch the video, do a neurologic test kind of asynchronously. And I said, let's do it in the clinic. I don't want to do this at home. So we'll have like someone sitting by at the waiting room. And it was kind of right at the bridge too far. It worked really well. You know, the production quality was high, higher than most. And that was probably a mistake. We spent a lot of time and we kind of failed. We probably should have started cheaper and done it faster and done something kind of more lightweight instead of, you know, this kind of really well-produced kind of video, etc. But at any rate, we learned kind of like how far to go. All right, almost done. How about data from repurposed devices? All right, so okay, maybe you've like done some home remodeling or insulation, and you think, well, do I need to like new windows? Well, what you do, contractors, they go buy a thermal camera, and they can kind of see where all the heat's leaking from around the windows, and it's really not that expensive. It's like $300-400. And you can get one that actually hooks onto an iPhone. Just looks like this. They're made by FLIR. So I had this crazy idea, like, you know, does everybody know what cellulitis is? Like skin infections, right? So in the olden days, like the modern world, you'd use like a marker and you draw around the redness, and then you date and timestamp it. And you use that to figure out, hey, is it, I hear some people are nodding. And it seems like so medieval. Like you actually draw a line and say, wow, did the redness go up or not? And that's the state of the art. So let's use a thermal camera. And we started doing this, and it turns out that it works really well. And so this is the same patient, just 2 days apart, and you can see the heat actually here, and you can see how, you know, there's the red line. You can see the heat. Here we have that washer ID again, because we scale it, because I can't really, it's really unstable. The breakthrough was being able to sort of size this and figure out how much the area is. Think of a topographic map. So we can actually, there's a surface that we can actually estimate, a heat surface. And then we can track that surface. And you can see this is just 2 days of therapy, right? It's really breaking up. I'll give you some other examples here. These are some other use cases. So this, a toe stab, you can easily see that, right? So, I mean, well, it doesn't look really great here, but here, there's just no blood flow. The toe's dead and needs to come off. And you can see that here as well. Here you can see it's red, they drew a line, but it turns out the redness goes way up and it's blocked by the pant leg. And so it actually, they put the marker here, the research assistant put the medical student put the marker here thinking it would be cold. And actually the heat was going up here. Here's a patient that has cellulitis in the arm, and it actually looks really bad when you first see him, but when you look at the heat surface, it's actually getting much better. And so a lot of times in ID, we get called, oh, come and see this patient. And Dr. Bridigan would know better, because he taught me lift the leg up, and then the redness goes away. Because the redness is a sign of the tissue damage from the past inflammation. But the heat is actually goes out beyond and then regresses. So it's like a leading indicator, and then it goes away. So it's a really nice way of seeing and sort of how someone's doing quickly. And well, what if you have dark skin? You can't even see the redness. And so here it's obviously red, but you couldn't see that from the photo or the surface. So this is an example of something, just a few $100s, wasn't made for health, but here's some real world data that you can use to help learn about patients. Okay. All right. What about getting students involved? We weren't the first person to do this, but it was a pretty easy student project. This is a Wii balance board. You can get these now. They're just being thrown away. Does everybody know what a Wii U is? You know, game board? Well, it turns out it's a pretty good balance board. And so we had an undergraduate student build a system where they use this as a force plate. And here you can see sort of what happens, the sort of tracing of the center of gravity of someone that's 24, 25, 83 that has good balance, and someone that's 83 that's fallen a lot. And so we took this to local nursing homes, and we enrolled a couple hundred patients, and we were actually able to show that this information alone actually did a pretty good job. We blinded it, and it was able to sort of suggest who was actually using an assist device. So we could tell the difference between people using walkers, canes, and not. And so this is like an example a student could do. Imagine using this in an emergency room or something like that. So I'll just give you one more kind of real world use case where you can take data, you can make your own real world data. It's a COVID example. Here we go. We've been doing stuff with badges lately to track movement between people in hospitals to figure out how infections spread. So right before COVID, you know, everybody thought this was kind of crazy, but people didn't afterwards. We went to the CDC, funded us to go to Atlanta and to distribute these small badges that talk to each other. And they don't actually talk, but they send small signals that the signals degrade over space or time and space. And so we can estimate distance. So we can see if people are within 6 feet of each other or 3 feet or 12 feet. And we handed this out to all the healthcare workers. We put them all in the rooms in these two very different nursing home configurations. It took us about an hour to set up using this hardware. This is sort of, we didn't make this, we bought these Nordic devices, badges, you know, I think they were like $25 each. We put them in sort of standard name tag badges. We bought some old cell phone batteries, made a badge. Then we actually put one of these in every patient room. We put them in these little boxes. We built it in the hotel. They didn't seem to mind in the lobby. And we built these boxes, put one in every single room. And from that, we were able to understand how different healthcare workers were connected in a nursing home. And the reason that's important is because we can understand how infections spread. So it's an example of real world data that you kind of go out and get. Up until this time, people have been using diaries or something like, hey, how many times does a nurse go into a patient's room and try to model infections based on this? But this is sort of just for 4 hours. We got about 197,000 messages. And from that, we can actually figure out where people are and whether they're interacting or not. And then we can actually study kind of patterns of flow. And right now, we're using this to figure out if there are other ways that we can redesign workflow to make it harder to spread infections in a nursing home or a hospital. At any rate, it takes kind of a team. We have computer scientists, we have bios, kind of the in red, the in blue, kind of our data scientists, kind of a biostatistician, and two sort of data scientists that were my wife's graduate students that we were lucky enough to retain at Iowa. We have an exercise physiologist. My wife's a health economist. And we've dozens of students. Like I said, every project actually involved a student or a resident. And we've had some funding, but a lot of this has been done just on a shoestring with pilot grants. And we didn't build a platform. Here's the project. And then we figured out what we needed to do the project. You know, we wanted to just be low cost, low impact, and move quickly. Anyway, that's about all. Thanks for your patience. Do you have any questions? Yes.
So this is more asking for advice as like an early investigator, and if you're interested in working with companies with biotech data specifically, how do you approach them and get in the door to get access to that data?
Well, I mean, the interesting thing is how companies want to work with you as long as it helps them. And then it's like, they're really excited, and then they sort of don't need you anymore. And,
Surprising.
you know, they're sort of like, there's one thing doing something in a clinical trial where there's a lot of money sloshing around to get, you know, during, you know, there's a vaccine trial or a big drug trial. And it's sort of harder to get their attention when you're just an individual researcher trying to get preliminary data. So I think what I would say is, what do you really need? And don't try to build something that's so elaborate. What is it that you need to get you the preliminary data to take the next step? Because I've been burned when I build something, when I build that thing that's just too big. And then I'm building the thing, I'm not doing the project. What do you need? What do you want to do? Why are you doing it? What is it that you really need? Go after that. I guess that's where it works. And where it doesn't work is when I spend too much time on the platform or the database.
Great. That's good advice. Thank you. Appreciate it.
There's a book, like a chapter in a famous computer science book. And it's like, throw the first one away. Like, cause you know, just try to build what you need.
Thanks for the talk. My question is just about if you witness any hesitancy in people sharing this data earlier it was talked about vaccine hesitancy and with all the conspiracy theories and distrust in healthcare. I'm just curious what your experience was with people sharing their data.
Well, everybody's consented. In general, I guess it depends on the study. I think the creepiest thing I've done is the badge project in hospitals. You know, that's a bit of a pitch, and we sort of make the public health goal. It takes a lot of work, but in general, when we went to Atlanta, everybody wore the badges. I mean, it is kind of big brotherish. We solved that by not getting more data than we need. Like, we don't learn more by doing like 6 days. We could learn almost everything we need in 4. We just do 4. We try not to get more than we need, and we try to be very transparent. In a project that I haven't talked about, we've used cameras in patient rooms because I actually want to study when a patient gets touched or not. Like, not just did someone go in a room, but actually where they went in the room. So we've been using Microsoft Connects to do that. And so there we built a system to show the nurses what was actually being collected. Like, this is all we're getting. It's a skeleton. You know, we're not saving, it's a camera, but we're just saving the skeleton. We don't record anything. So we try to be very transparent, tell people what we're actually collecting. I don't know if that makes sense, but it is an issue. And we try not to ever ask for more than we need.
I guess my follow-up question is, do you think there's sample bias in those people who do consent versus to those?
Well, we were concerned about it in the healthcare facilities, but we got everybody to wear it. So we really haven't had that problem. In other cases, you know, we had really good luck with blood pressure. People are really, really interested in their own blood pressure. And that's been really successful. Other cases, I'm doing a project right now where funded by the CDC, where I'm trying to track C. diff. And so I'm asking people if they have diarrhea at home, and they're pretty sick people. And they're usually, when they don't do it, it's usually because they just don't feel good. And you know, they don't want to do that. They want to get better. And so it's like, in my experience, it's been usually more onerous, or sort of, it hasn't been really the privacy issue. But I think if studying other things, it might be.
Thank you.
Fascinating. I'm thinking of 2 applications that I would have used or would use in the toxicology world with envenomation, specifically snake envenomation and brown recluse, oxalicism, because I have to talk many surgeons that are doing fasciotomies for snake bites. But the other issue is circumference. We're still marking, back in the archaic days of marking. We're still in the archaic days of marking circumference. And I don't know if there's anything to actually replicate that digitally. But with brown recluse, if you're worried about ischemia, you might have zones of hot and cool. And it takes my eyes to look at it to make a diagnosis. And of course, when I publish on it, the journals always say presumed.
Right.
That's
a great idea. So you're worried, you think that actually might change the perfusion of the skin, the bite. We can see abscesses.
Parkinson's syndrome, you're losing
Yeah.
blood flow. It looks terrible.
Yeah.
In my experience, it's suprafascial. It's above the fascia, unless you just happen to be the most unlucky person that got a snake to go through your fascia, which is pretty uncommon.
So most people who got bit by a snake know they've gotten bit by a snake, and so, you know. Uh-huh. Mhm.
But something like that to show the surgical specialty.
Like that it's not growing, for example.
You don't need to.
Yeah.
It's really swollen. It's really swollen, but it's not.
But it's
causing perfusion abnormality.
I see. That's true. So I can tell you right now that one of the most exciting use cases is necrotizing fasciitis. I have some pictures I cannot show about a disease called Fournier's gangrene, a special case of necrotizing fasciitis. Don't look that up at work or at home. We published it in the Journal of Urology, but that's when the tissue is actually dying. And it looks like this works, and you do need to do surgery. So we're very excited about that use case as well. So I hadn't thought about snake bites.
It's another crazy idea for you, but think about it.
Yeah, I
elaborate on that.
We'd love to collaborate. In fact, that's why Matt asked me to come. Maybe, I don't know. Is that?
I hope so.
Yeah.
Really interesting. I'm just curious, how is it that you could get the thermometer data from digital thermometers?
From, okay, the thermometer data from the Bluetooth company,
Yeah,
they gave it to me because I'd written these other papers and I spent time talking to the guy and it was great, but I didn't get a grant, but it was helpful. It was a good journey. But the problem is that once he didn't need us and we wrote this paper in clinical infectious diseases, right? He doesn't need us anymore. And we no longer have a relationship.
so sound.
But and I think that's sort of, that happens a lot. I'm sure I'm not the only one that, and I don't have, it's not like I'm crying at home or anything. Oh, it was fine. I had a choice. We did this. It was like a, transaction and I'm happy. But it's hard to get, to get device data from like 500,000 people.
Yeah. I'm thinking more logistically, and I just am not.
Logistically, so they,
Someone takes your temperature and then.
so they were selling this as a business. They were selling thermometers to mothers.
Right.
And their use case, they were trying to sell ads and work with CVS and children's hospitals and sell ads. I don't know. That wasn't my, like,
Right.
I would have done their job differently. Like, but that wasn't my
Mhm.
job. I didn't get in their business and tell them what they should do. But they were convinced of this. And I said, well, I think this can be used to predict a disease. I think this could be used to understand something we didn't really know because of the scale. Like, you can get 10 million readings at University of Nebraska, but you can't get the readings at home before they get to University of Nebraska.
Did the mothers know their temperatures were being?
So it's all the data that we got was de-identified. But in the that terms of use for everything, they clicked on it saying that they would send everything out there. But we only got like zip code level data
Right.
and age. So there was nothing that was identifiable.
Got it. Thanks.
So
Like your crazy idea.
yeah, there was a very interesting conversation with like a reporter at Wired that kept asking these questions, like these gotcha questions. And I said, well, we don't have any data about people. We just have, geographic and timestamp data. So, but I think, my 2 cents is that anybody in this room can do these projects and this data is out there. And, I think the question comes first and then just figure out how to get the data.
Thank you, Phil. That was fabulous. Brett, you did a good job.